Welcome, everyone. Today we're taking a journey into the world of DNA extraction, a technique with rich history that has evolved over time. So let's embark on this exciting adventure together. DNA extraction is a fundamental technique in molecular biology that involves isolating and purifying DNA from biological samples such as cells, tissues, or body fluids. The goal is to obtain a pure and concentrated DNA sample free from contaminants and other cellular components. The purified DNA can be used in various downstream applications, including genetic testing, research, and forensic analysis. Now, let's take a closer look at the working principle of DNA extraction. The working principle of DNA extraction revolves around isolating and purifying DNA from biological samples while maintaining its integrity and stability. This process typically involves three main steps. The first step is cell lysis. It is to break open the cells or biological samples to release the DNA. This can be achieved through physical methods by applying mechanical forces or energy to disrupt cell structures, including the cell membrane and nuclear envelope. Some common physical methods include grinding, vortexing, sonication, freeze-thaw cycles, and French press. Physical methods can be effective for various sample types, but they may also cause shearing or fragmentation of the DNA, which might be undesirable for some downstream applications. In such cases, chemical or enzymatic methods, which use specific chemicals or reagents to disrupt cell structures, might be more suitable. Some common chemical methods involve use of detergents, chaotropic agents, enzymes, and alkaline lysis. Chemical methods can be effective for various sample types, and they may be combined with physical or enzymatic methods to enhance cell lysis efficiency. However, they can also introduce contaminants, such as proteins or residual chemicals, that may need to be removed during the subsequent DNA purification steps. After cell lysis, the DNA needs to be separated from other cellular components such as proteins, lipids, and RNA. This step is called DNA purification. Various techniques can be employed to achieve this separation, including organic solvent extraction, for example, phenylchloroform, solid phase extraction, for example, silica-based spin columns or magnetic beads, or enzyme-based extraction, for example, salting out. These methods exploit differences in the properties of DNA and contaminants, such as solubility, charge, or size, to selectively isolate DNA. When choosing a DNA purification method, several factors need to be considered to ensure the desired purity and quality of the extracted DNA. Sample type, downstream applications, DNA fragment size, yield and purity, etc. The final step is DNA concentration and storage. Once the DNA is purified, it may need to be concentrated depending on the desired downstream application. This can be achieved using various methods such as alcohol precipitation, for example ethanol or isopropanol, vacuum concentration or speed vacuum, microconcentrators or ultrafiltration, and dialysis, depending on the specific requirements and desired outcomes. The purified and concentrated DNA is then resuspended in an appropriate buffer, such as TE buffer or nuclease-free water, and stored following proper guidelines. For short-term storage, DNA can be kept at 4 degrees Celsius in a refrigerator, while for long-term storage, it is recommended to store DNA at -20 degrees Celsius or -80 degrees Celsius in a freezer. Aliquoting and proper labeling of the samples, as well as maintaining a record or a database, will further ensure the integrity and usability of the DNA for future experiments and analyses. Various platforms are available for DNA extraction, ranging from manual methods to automated systems. The choice of platform depends on factors like sample size, throughput, and budget. In this video, we will only be discussing manual methods and automated systems. For manual methods, these are low-cost, hands-on techniques that typically involve using basic laboratory equipment and reagents like centrifuges and chemicals. There are several commercial DNA extraction kits available in the market that utilize manual methods for DNA extraction. These kits often come with pre-formulated reagents and step-by-step protocols to streamline the extraction process. Some popular manual DNA extraction kits include the Thermo Fisher Scientific GeneJet Genomic DNA Purification Kit. This kit utilizes a silica membrane-based spin column technology to selectively bind DNA, ensuring that the purified DNA is free from proteins, nucleases, and other impurities. The protocol involves several steps, including cell lysis, DNA binding to the silica membrane, washing to remove impurities, and elution of purified DNA. It is designed for various sample types, including cultured cells, tissues, blood, bacteria, and yeast. The Monarch HMW high molecular weight DNA extraction kits are designed to isolate high molecular weight DNA from various sample types, including cultured cells, tissues, bacteria, and blood. The protocol involves several steps, including cell lysis, binding of DNA to magnetic beads, washing to remove impurities, and elution of purified high molecular weight DNA. These kits are particularly useful for applications that require long DNA fragments, such as long-read sequencing technologies like Oxford Nanopore and Pacific Biosciences or PacBio sequencing platforms. The ProMega Wizard Genomic DNA Purification Kit. The kit is based on a salting out extraction method, which allows for the effective purification of DNA without the need for organic solvents or hazardous chemicals like phenol and chloroform. The protocol involves several steps, including cell lysis, protein precipitation, DNA precipitation, washing to remove impurities, and rehydration of purified DNA. It is a versatile system designed for isolating genomic DNA from various sample types, such as cultured cells, tissues, whole blood, and bacteria. BioRad Instagene Matrix. This product is based on a Chelex resin, which binds to and effectively removes metal ions and other cellular contaminants while leaving the DNA in solution. The protocol involves several steps, including sample lysis, incubation with Instagene Matrix, and heat treatment. Then the supernatant containing the purified DNA is collected for downstream applications. The Chelex-based method is particularly useful for extracting DNA from small samples or when a rapid extraction is required. Fully automated DNA extraction systems are designed to streamline and simplify the process of extracting DNA from various sample types. They work by automating the various steps involved in extracting DNA from samples. They use robotic arms, liquid handling systems, heating blocks, and other integrated components to transfer samples, mix reagents, and perform the required incubations and separations. Fully automated DNA extraction systems are essential for a high-throughput applications, reducing human error and ensuring consistent results. Several fully automated DNA extraction systems are available on the market, each with their unique features and benefits. Here are a few notable systems. QIAGEN QIAcube Connect. The QIAcube Connect is a compact, fully automated system that uses QIAGEN's spin column technology to extract DNA from various sample types. It can process up to 12 samples simultaneously and offers pre-installed protocols for a wide range of applications. The Thermo Fisher Scientific Kingfisher Flex. The Kingfisher Flex is a versatile high-throughput system that uses magnetic particle processing technology to extract DNA from various sample types. It can accommodate 24, 48, or 96 samples per run and is compatible with Thermo Fisher Scientific's range of extraction kits. ProMega Maxwell RSC. The Maxwell RSC is a compact benchtop system that uses paramagnetic particle technology to extract DNA from various sample types. It can process up to 16 samples simultaneously and is compatible with a wide range of ProMega's DNA extraction kits. These fully automated systems offer advantages in terms of speed, throughput, and consistency, making them suitable for high-volume laboratories or those with stringent quality control requirements. When selecting a platform, it's essential to consider factors such as sample type, throughput, budget, and the intended downstream applications. In conclusion, DNA extraction is a fascinating and vital process that unveils the genetic blueprint of life. By isolating DNA from various samples, we gain insights into our health and the world around us. Thank you for watching.